In this video, we will discuss about the primary and secondary response of our immune system. So if you look at this particular diagram, you can see that a human body has been surrounded by lots of viruses. Similarly, in our environment, we are surrounded by lots of microorganisms which are pathogenic. So this pathogenic microorganism always try to enter in our body system. And what happens when they enter in our body system, they will try to harm us or they might lead to our death. So our body has evolved mechanism that will help us to get rid of this pathogenic microorganism. So there are systems that help to remove this pathogenic microorganism from our body, and that system is what we called as immune system. So if you look at this slide, you can see that we have both innate immunity as well as adaptive immunity. So I hope you guys know the meaning of immunity. Immunity is the defense against microorganism. So innate immunity comprises of this epithelial barrier, dendritic cell, neutrophil, macrophage. Okay, so all these cells, they provide innate immunity, and they are the first line of defense against microorganism. So whenever our body encounters any antigen, in that case the first defense that our body will play an important role is this innate immunity. This innate immunity provides the first line of defense, and they start within a few hours from 6 to 12. Okay, so their activity starts within hours, and they will try to remove the microorganism from our body. However, if this immune system gets failed to remove that particular pathogen microorganism, in that case this adaptive immunity will come into play, and the response that is generated by this adaptive immunity, it is very slow when you will compare it with respect to innate immunity. The innate immunity will start its activity within hours, as you can see, but this adaptive immunity, it will take few days. Okay, so this is the difference, basic difference between adaptive and innate immunity. So this adaptive immune system, it comprises of the T cells and B cells. B cells basically is mediated by the serum antibodies, and T cells, they secrete out certain chemicals such as perforins, granzymes that help to kill any foreign particles. So both this innate and adaptive immunity, both of them plays a crucial role in the removal of pathogenic microorganism, as well as help our body system to fight out any pathogenic microorganism, because it is very necessary to remove this pathogenic microorganism. We are always exposed to pathogenic microorganism in our environment, and we have a 100% chance of encountering or taking them inside our body anytime. So it is very important for our immune system to remain boosted up, to remain in an active state, so that we can deal with such pathogenic microorganism. So if you look at this graph indicates the primary and secondary response of our body to any antigen. So let me at first describe you what is primary response. So primary response is the response of our body whenever we are first exposed to any antigen. That means, let us suppose a virus, a pathogenic virus X enters in our body. So this virus is entering our body for the first time. So whenever this X enters in our body, so that is the primary response. The body will generate an immune response against this X with the help of either innate or adaptive. That response will be known as the primary response. Okay, so the first exposure, our first exposure to an immunogen, to an antigen, that is known as the primary response. So during the primary response, what you can see in this graph, there are four different phases that will occur. The first is the lag phase. So in this lag phase, what happens, there is no production of antibody. So I have told you that in this adaptive immunity, the antibodies, the B cell mediated process that involve our serum antibody, plays a very important role. So during this lag phase, there is no production of antibody. Okay, so there are four different phases in this primary response. So the first comes this lag phase, and in this lag phase there is no production of antibody. However, and in this case also the B cells remain naive. What is the meaning of naive? Naive means the B cell has not encountered any antigen. In that case it is an immature B cell. Whenever B cell does not get exposed to any of antigen, in that case such B cells tend to be known as naive B cells. So in this lag phase, the B cells generally before the start of this lag phase, the B cells remain in a naive condition, in an immature condition. But whenever these B cells encounter this antigen, it will start activated. Okay, this B cell will get activated, and then it will lead to clonal expansion of that B cell that will ultimately leads to production of antibody. So how this B cell gets activated, I have already discussed in another video. So once this naive B cell will encounter antigen, it will start differentiating. Okay, and when it start differentiating, it will then leads to production of antibodies. So this lag phase, when where there is no antibody production, it is followed by log phase. Okay, so in lag phase, LAG, there is no production of antibody, and this lag phase is followed by log phase. So what happen in this log phase? In this log phase there is production of antibody. So what happens? The antibody gets produced, the antibody will be produced, and there will be an increase in the concentration of antibody in our blood. Okay, so when this concentration of antibody will increase, what this antibody will do now, they will go and bind this antigen. So you can see here, this is antigen, this antigen is bounded by this antibody. And an important thing that happens during this primary phase, primary immune response, is that the major antibody that is produced here is IgM. Okay, so IgM being pentameric, you can see this is pentameric structure. They will form pentameric structure and they will start binding. So once this IgM pentameric complex will bind antigen, they will start getting removed from our body. Both this antigen antibody complex will be removed from our body, and in this case you will see the decline phase. That means the concentration of antibody is getting decreased. Why? Because both antigen and antibody are getting removed from our body. So this axis, this is the axis that denotes the concentration of antibody. So if you will go towards upper side, it will indicate that the concentration of antibody is increasing. So as you can see here, during the log phase this curve is moving towards the upper side, and when this antibody forms complex with antigen, the body will remove this antigen antibody complex, and there will be a decrease in the concentration of antibody in our serum, and you will see that this graph or this curve will be falling towards the downward side, which means that the concentration of antibody gets decreased. So in this primary response, which takes around 14 days, the production of antibody takes around 14 days, takes a lot of time, but it will remove the antigen, and the production of antibody is also very less in this primary antibody response. Now once you can see that the concentration of antibody is getting declining after this steady phase. Okay, after this steady phase you will see that the concentration of antibody is getting declined. Then what? So the beauty of this adaptive immune system is the memory response, which is not present in the innate immunity. Adaptive immunity consists of memory response. So whenever our body is exposed to the same antigen again, in that case what will happen, the memory cells will come into play. Okay, so again if this X, as I have told you that during the primary response the antibody will bind to this X and they will remove it from our body. But what happens, these X are present in large amount outside our body, they are present in environment, so we have chances that we will encounter the same X again. There is again chance that this X might enter in our body. So what happens in that case? So as I have told you that the beauty of this adaptive immune system is the memory response, the memory cells that are present. So whenever this X again enters in our body, the memory cells will come into the play. Okay, so in this case what you will find that there will be a very short lag phase compared to this primary response. Okay, but once this short lag phase is over, the log phase is very large compared to your primary immune response. That indicates that the concentration of antibody, the amount of antibody that is produced in the secondary immune response is very high. And once this antibody gets produced, it will not take a longer time as primary antibody response has taken, which took almost about 14 days. But in this secondary antibody response, it will take from 3 to 5 days for the production of antibody. So the amount of antibody that is produced here is very high. And why it is taking 3 to 5 days? Because these are the memory cells. So when these B cells divide, they divide into plasma cells, B cells, or memory cells, M cells. Okay, so in the primary response, this M cell was also produced, but they were stored. Why? Because when the body will face similar antigen again, when they will encounter this X again, in that case this M cell will fight. Okay, so that's why it took around 3 to 5 days, very lesser time compared to the primary antibody response. And also the concentration is also very high. The memory cells are produced in huge amount and they are stored, and whenever they will again encounter, they will start doing their job. So the concentration is very high, and there is a sharp increase in this hill of in this antibody concentration range, and again they will bind. But unlike primary antibody response, where IgM was majorly produced, in the secondary immune response, IgG is produced in large amount. Okay, so IgG is the major antibody that is produced during secondary antibody response, and IgM is the major antibody that is produced during primary antibody response. So if you look here, in this response IgG is produced, and once this IgG completely binds the antigen, it will again, both this antigen antibody will be removed. They will completely bind the antigen and completely remove it, and then there is a decrease in the curve of antibody. The concentration of antibody in the serum will decrease because some of the antibody that has been produced will get eliminated with antigen. Okay, but since the concentration of antibody during secondary antibody response is very high, so we will not observe a very sharp decline in this antibody graph which we have noticed in the primary response. Okay, so the concentration of antibody was very low and the decline was very sharp. However, the decline is not very sharp. So in this case, the memory cells that are produced, they have a very long life. They are produced in very large amount. They are produced in very short times. Okay, and the major antibody that are produced during secondary antibody response is IgG. However, when you will look at the primary antibody response, in this case it will take large time. The antibody production is very low. The major antibody that is produced is IgM, and they are very short-lived compared to secondary antibody response, where the plasma cells have a very long life. But in case of primary antibody response, these plasma cells have very short lives. So I hope you have understood the concept of this primary and secondary antibody response. Put your question in the comment section. Thank you for watching.